Hi everyone, my name is Vanessa, and today I will be sharing the results of my investigation into the metapopulation structure of the California mussel. The California mussel inhabits the coastal waters of western North America, from Baja California to Alaska. This intertidal species spawns year-round, a process after which the larvae drift in the water column for up to 45 days. The mussels are also long-lived, up to 50 years. These traits contribute to a very large population size, and together these are just a few of the factors that influence the population structure of the species. The focus of this project is to quantify the amount of genetic variation between subpopulations and to search for evidence of population structure. A metapopulation is composed of spatially separate populations that exchange genetic material to varying degrees with one another. Understanding how often and in what direction metapopulations exchange genes provides insight into the demographic history of an organism. This is relevant for decision-making and conservation efforts and for broader evolutionary inference. We can think of a metapopulation using the serial dilution analogy. If we start with an allele in Northern California, it is going to be passed with high frequency to its neighboring population, but it will be diluted, so to speak, as it progressively is passed to populations further south. Because of this, we expect allele frequencies to change subtly over space, just as we expect concentrations to change subtly between dilutions in a serial dilution. This is the premise of isolation by distance, which reasons that because we expect less gene flow over greater geographic distances, we also expect higher FST over greater geographic distances. FST is the statistic that measures the amount of genetic variation that is partitioned among populations. In two populations with equal amounts of two haplotypes, the FST will be 0, indicating no structure. If these two populations are distinct in that one entirely consists of one haplotype and the other exhibits the second haplotype, then FST will equal 1, which indicates complete structure. In this study, we use phi-ST, which is a variant of FST used for sequence data. Previous research has found the California mussel to be genetically homogeneous throughout its range from Baja California to Alaska. These studies find low phi-ST, or a lack of population structure in the California mussel. Notably, Kelly and Palumbi found only 13 of 50 intertidal species to show significant population structure using statistics. These studies cite high rates of gene flow and lack of selective pressures as potential reasons for the observed lack of population structure. Given that FST and gene flow have a non-linear relationship, and that FST is poor at detecting structure when migrants exceed 10 per generation, this project aims to recapitulate previous results with the expectation that FST is not a sensitive enough statistic to detect population structure that may be subtle but meaningful. To achieve this, I isolated mitochondrial DNA from the veal tissue of 138 mussels, calculated multiple diversity statistics, and tested the hypotheses that genetic partitioning occurs at either point conception or gate minuscule. These are geographic projections which significantly alter coastal currents and upwelling, and therefore may impact larval dispersal and gene flow. In addition, I acquired 152 sequences from NCBI. These sequences represent 6 populations in the Pacific Northwest. Table 1 is a summary of diversity statistics broken down by population. Drawing our attention to the nucleotide diversity results, we can see that there is a relatively low amount of diversity at the nucleotide level. Nucleotide diversity is the average diversity per nucleotide. In other words, if we were to pick a random position from 2 random sequences, this would be the probability that they would be different bases. Looking at haplotype diversity, these values are much higher, and that's because this measures how unique any given haplotype is in this dataset. A haplotype is a set of alleles that an organism inherits from one parent. This will naturally be more diverse. Looking at figure 1, we have a haplotype network. A haplotype is a set of alleles that an organism inherits from one parent, and a haplotype network visualizes the relationships between sequences. Here we see 150 haplotypes that are relatively similar to one another. The larger nodes indicate that more samples belong to that haplotype, and the colored slices indicate the relative proportion of each population that constitute the haplotype. The segments indicate the number of base substitutions required to get from one haplotype to the next. Here I plotted the inverse function of the distance between pairs of populations as a function of linear distance. These plots indicate a decrease in FST and phi-ST scores as distance increases. This conflicts with the expected result, which would show an increase in FST or phi-ST, which is analogous to a decrease in gene flow with distance. I conducted an analysis of molecular variance to test for partitioning of genetic variation at point conception and Cape Mendocino. These hypotheses did not yield significant results. In addition, I calculated the overall distance between all populations for the purpose of comparison with previous studies. The results are consistent in failing to detect population structure. In summary, our results are mostly consistent with previous studies that failed to detect population structure using traditional methods. However, the presence of high pairwise bias T values in our study are not consistent with previous studies. Although our AMOVA results do not suggest population structure. Population structure may be faint but significant. This warrants further investigation utilizing more sensitive methods of inference. In terms of future directions, coalescent samplers have greater sensitivity for detecting population structure when gene flow is less than 100 migrants per generation. Looking at the allele frequencies of the two populations in this example, we might consider the FST to be 0, but a coalescent sampler takes the genealogy into account, and it reconstructs the ancestral population looking back in time. For instance, since the red alleles are present in both populations, we assume that it was present in a common ancestor of the two populations. If population 2 is most likely at the base of the tree, we can infer that there was gene flow from population 2 to 1, which is how red alleles were introduced into population 1. In this way, we can make meaningful connectivity hypotheses and then choose the most likely model of gene flow using Migrate-N, a program for inferring effective population sizes and gene flow rates and direction within a metapopulation using a Markov chain Monte Carlo search of parameter space with coalescent simulations. And that is all. I would like to give a big thank you to Dr. Crandall, Dr. Ju, and Dr. Kanner for providing unparalleled mentorship. I would also like to thank UROC for all of the resources they provide to undergraduate researchers here at CSUMB. And I would like to thank the BD2K cohort of 2019, Charles Nye, Avery Calhoun, Lily Creer, and the G Lab for friendship and community, all of which I have found to be essential to the scientific